Good day everyone, my name is Nicholas C. Durgadeen and today I'll be presenting my summer research that I did at the Nature Conservancy this summer. The name of my mentor is Dr. Robin T. Smith and the name of my project is The Effects of Sahara Dust on the Growth of Juvenile and Adult Corals. Alright, so allow me to give you some background information on what the Sahara Dust is and where it comes from. So, the Sahara Dust is dust that travels all the way from North Africa, the Sahara Desert, and then spreads all across the Atlantic Ocean and in the Caribbean Sea. This has led some researchers, like Dr. Garrison, to believe that maybe the dust on its way traveling across the ocean has accumulated some sort of minerals that may have harmful effects on the ecosystems that we have. For example, the coral reefs. In fact, what we do know is that the Sahara dust is actually linked to the red tide bloom that happens off the coast of Florida. Now this has made me theorize as whether or not the Sahara dust may have an impact on the growth rate of stony corals. Okay, so allow me to be a little bit more specific about what exactly my hypothesis is. So, I hypothesize that the corals incubated in the water treated with Sahara dust are going to grow slower than the corals in the water without the Sahara dust, and that the coral polyps in the water with Sahara dust will not bud, whereas the coral polyps in the water without Sahara dust will bud. And in addition to that, I also recorded the reactions of a separate colony of coral polyps when they were exposed to the dust. I have to thank my mentor, Dr. Robin Smith, and the TNC because they gave me the liberty to come up with my own research project, if feasible. So prior to my summer internship, the Virgin Islands experienced this largest Sahara dust bloom that it has ever experienced in decades. This gave me the idea for the project. All of the dust collected and used for this project was collected immediately after and during the Sahara dust bloom we were experiencing. To find out how much dust the Virgin Islands was exposed to during the Sahara dust bloom, I used a website called earthschool.net, which gave me the approximation of how much dust was in the air within a unit of area. Then conversions were done to gauge how much dust to dose the two miniature-sized tanks, and then the number we derived was 0.02 grams. On the left-hand side of the PowerPoint, you can see the two species of microfragments that we used, the Orbicella fabulata and the Diploria labyrinthiformis. To be exact, the OFAV was the coral microfragments that you see on the left-hand side of your PowerPoint, and the D-lab, that was the coral polyps you saw in the first initial slide. So, I also mentioned that I used the D-lab, the coral polyps, and was trying to determine their response and reaction after being exposed to the Sahara dust bloom. For that, I simply dispersed the dust onto the surface of the water while looking at the corals under a microscope. Initially, the corals that were incubated in salt water treated with Saharan dust weighed more than the corals incubated in salt water without Saharan dust. However, the difference between the weight was not as large as it was initially. In week 1 of 4, the difference in weight was only by 0.056 grams, whereas the final difference in weight was by 0.146 grams. Even when you compare the weight difference for each category, the Sahara dust treatment and the non-Sahara dust treatment, to their own initial weight, the difference in weight is still noticeable. The difference in weight between initial weight and the final weight is 0.17 grams, whereas the difference between the week 1 and week 4 for the Sahara dust corals is by 1.07 grams. That's a whole 0.9 gram difference. With this data, I was able to calculate how much each week the corals in the non-Sahara salt water increased. So, in the non-Sahara salt water, there was an increase by 0.042 grams, whereas each week in the corals that were in the Sahara dust water increased by 0.26 grams. In addition to this, none of the 7 coral polyps in the Saharan salt water underwent the budding process. Yet, in the water without the Sahara dust, 2 out of 5 of the corals underwent budding. Also, the coral polyps exposed to Sahara dust took on average 121 more seconds to re-extend their tentacles after being exposed to the Sahara dust. My mentor from this information proposed an article to me by Dr. Rich, where he discussed mineral enrichment and specifically iron, and how corals are impacted by the depletion of specific trace minerals such as iron, zinc, magnesium. So for the discussion, I'll simply be talking about my experiences and things I would have liked to be able to do or could see myself doing in the future. So the first part of my experience was with WAPA, the Water and Power Authority for the Virgin Islands. WAPA was having a series of power outages during the beginning and end of my project. Corals are very temperature-sensitive animals, so there were lights placed above them and then a cooler for the water to keep them at a relatively safe temperature, 28 degrees Celsius to be exact. I don't believe the change in temperature actually had any sort of impact on the data, simply due to the power outages not causing any sort of drastic changes in the water temperature. Another minor inconvenience was the lack of equipment I was able to use. I would have liked to be able to measure the composition of the dust so that I could then determine the exact minerals within the dust that could have helped promote the growth of the corals. I also had some difficulty finding any sort of literature on the specific species of corals I was using, like the OFAV and the DLAB. So I wasn't able to figure out when exactly the corals were going to undergo the budding process. So what I did instead was I simply used information from similar coral species and then used that as a sort of reference. And prior to the experiment, I actually found that the salt water that the corals were living in were actually artificial, meaning that they weren't taken directly from the ocean. I believe this aids the idea that the water that the corals were in were lacking in certain minerals that would have helped promote their growth. Minerals such as magnesium and calcium. If I had more time and resources for the future, I would have liked to be able to see myself collecting dust from different parts of the Caribbean, and then also measuring the composition of the dust to determine whether or not it actually contained growth-enhancing minerals. So to conclude, I do believe that the minerals within the Sahara dust did influence the coral's calcification growth. However, that was only due to specific conditions being met. One of those conditions is that the corals, I believe, were lacking certain nutrients that they would have received from the ocean. So I don't believe that at a larger scale, the Sahara dust bloom actually did influence the coral reefs and had any sort of negative impact on them. But none of the coral polyps did actually bud in the water treated with the Sahara dust, whereas in the water without the Sahara dust, there were coral polyps budding. This again, I don't believe would show similar results if repeated on a larger scale, like within the US Virgin Islands, much less the entire Caribbean. Here are my references. One is of the paper I mentioned earlier, the one written by Dr. Garrison, who proposed that corals are being negatively impacted by the Sahara dust. And the next is on mineral enrichment by Dr. Reich and others, that I mentioned in the results. For my acknowledgements, I'd like to thank specifically Dr. Robin Smith and the Nature Conservancy for allowing me to come up with my own project idea. I'd also like to thank the University of the Virgin Islands, Emerging Caribbean Scientist Program, and the Seas Your Tomorrow program, led by Dr. Kristen Grimes. And then, of course, the National Science Foundation,